I think I sort of explained to you yesterday sort of my background with this laser microdissection. I had only been in Shreveport about 6 months, having come from the New York City lab, which is, very large volume type of casework lab. And, I remember immediately the first thing, it was like, oh, we're doing organic extractions. And this was on, blood, blood samples, differentials. We were doing a chi-amp for references, which is a little quicker, but I thought, oh my gosh, it's going to take forever for me to do. I'm doing my casework from beginning to end. How am I going to do this? And so in my head, I'm thinking, how can I batch things? And, so anyway, 2004, thank goodness I saw Christine speak at the American Academy meeting, and immediately I was blown away. I was like, oh my gosh, And so I went back to the lab, and I was actually, maybe it was that first week back, and I had asked Christine some questions afterwards, and, first week back I was really excited about it, but didn't necessarily voice that to my boss, because I was sort of new still. I didn't just want to be like, oh, we need to change this, let's, And actually that same week, I think he came up to me and was like, Kelly, you were at the meeting, did you hear anything about laser microdissection? I said, actually, I'm so glad you asked. I went to this presentation, it was fantastic, and I can't believe this, and I think it was just that summer in 2004, we had a DNA meeting on how to spend the capacity enhancement money. And so we, let me find the mouse here. We were trying to figure out how are we going to spend that money. I don't know why this won't go. There we go. Seems really slow. Anyway, so we were trying to decide how are we going to spend this money. And of course, immediately in our heads, we said, well, what are some of the bottlenecks in the laboratory? And I don't know why I can't get this to work. Does anyone?
Use the keys. I had to switch to page up and page down keys.
Okay. So we said, well, we have to identify bottlenecks in the laboratory, but I'm getting ahead of myself here for a minute. Well, first, let me talk to you. Today, the reason I'm here is because Christine and I, of course, when we first got this microscope, Christine was already doing work. And being that I already work in a case-working laboratory, and all I wanted to do, I didn't want to reinvent the wheel. So I called her up and I said, okay, can you give me some tips? Can you give me some pointers on how to maybe get started, and what can I do? And of course, she was doing her research project at the time under, her professor, and she didn't want to indulge everything, of course. So I said, that's fine. And so we kind of started off on similar paths, but then we kind of, veered off. So the procedure that she's gone through, this yesterday and today, is different than what I ended up doing, because I kind of got started and she gave me some guidance, and then we just completely veered off. So that's kind of what my talk is going to be about, and how we hope to implement this into casework. So some of the highlights, of course, is why, and I've already sort of gone into that. I'm going to touch just on how we decided that we were going to eliminate the manual extraction, and a little bit about reproducibility and sensitivity of that, that procedure, some troubleshooting I had to do, and then how I got into doing a single amplification and the optimization of the amplification, what we're going to do in the absence of sperm or no sperm, or a little bit of sperm, and then I'm just going to touch on the streamlined novel process of LMD that we're planning on doing in our lab, but Pat's going to go into more detail on that. Here's our microscope. This is the Leica AS LMD, and this is the predecessor to the one that you see in the laboratory. And actually, it's a great microscope. We've had it now since, I think November of 2004 we got ours. So we've had it now almost 3 years, and the laser still seems to be working great and everything. So anyway, why laser microdissection? Well, as I hinted to, one of the bottlenecks in our laboratory we immediately identified was look. A lot of our cases in the DNA laboratory, forensic DNA laboratory, are sexual assault type cases, sexual offenses, and that actually makes up about 45% of our cases. And of course, that translates into a good number of differentials. Given if you have, let's say, 40% of your cases are positive, that's a lot of differentials. So we wanted to come up with something. I said, look, we either have to, come up with a new way of doing the differential or whatever. Of course, I threw out the LMD because I was so excited. I'm like, oh, what about this laser microdissection? Well, little did I know, I didn't know Pat that well. And he loves research, so he was like, yeah, let's do that. And I mean, just within a couple months, he wanted to buy this microscope, and I was freaked out because I'm like, okay, I've never done a research project in my life, and I've never done a validation. So I was like, what? I don't know. I was scared, you know. But anyway, he said, oh, we'll be fine. Let's do this. I said, okay. So we got the microscope. Some of the disadvantages right off the bat is, of course, the initial cost. You know, everyone's kind of been talking, oh, you know, how are we going to buy this microscope? Do we really want to buy the microscope? Well, of course, back in the days when we were doing PCR and new genotyping, you know, you had to buy the 377s, even the 373s, you know, and now, of course, the 3130s. Those are pretty expensive. So maybe if this eventually becomes a mainstream thing, you know, it won't be a problem to get money. Of course, I had to do some type of novel validation for this procedure. The pen slides, like we talked about, were about $4.25 each. I think that price has gone up just a little because, like Andy was saying, now they're about $4.60 a piece. We've already talked about the architecture. You know, the pen foil contains these pores, which, you know, can have a tendency to be sort of difficult to see sperm, but of course it's a learning curve. And now, because I do this all the time, it doesn't even bother me to see these slides. One nice thing, at least about the Leica, except for these pen slides, everything else that I'm doing with this procedure, there's no other consumables. Everything else is already in your laboratory. So that's one of the nice things. Yes, the cost of the instrument. Yes, the cost of the slides. But, you know, otherwise, there's no other consumables. One other disadvantage I didn't put on here, and I don't know why I didn't, was if you have multiple sperm donors, this isn't going to help you. So, okay, a couple advantages, which we've talked about a bit already. One of the things is, well, maybe we can, well, the biggest one, of course, is the absolute separation of sperm and epithelial DNA. You know, hopefully by just being able to look at the slide, identify the two different cell types, we can cut them out, physically separate them now, rather than having to go through that whole chemical process. And then, of course, we were hoping we could eliminate our traditional extraction with this microscope. Maybe we could come up with a way to just completely eliminate that traditional extraction. These other two I'm going to go into later. So first thing that started, you know, going in my head was, okay, we've got this microscope. You put these little tubes, you know, PCR tubes on there to catch the sperm, but how are we going to get from, you know, point A to point B, you know, from this tiny tube? I mean, are we going to do an extraction? Are we going to just try to go right to amplification, you know, without much manipulation? What can we do with just this little tube? So we said, what would happen if we just directly amplify? I know that sounds crazy, but what would happen? So let's try it. Well, it didn't work very well. I mean, we did do amplification, and believe it or not, we did get peaks. We even increased cycles, you know, said, well, what happens if we bump up the cycles? And we did get peaks again, but what would start to happen, of course, just as you might guess, is we started to get artifactual peaks we couldn't explain, you know. We would get a basic, you know, the type, but then sometimes we just get this peak. Okay, you can't explain it. Okay, that's not good. We don't want, and we couldn't get full profiles. So the next thing was, well, because Christine, you know, had kind of talked to me about her use with Lysengo. Well, maybe we can do a preamplification lysis in the tube and then directly amplify from there. Well, there are some constraints. Number one, you don't want it to adversely affect your downstream PCR. And number two, you have a limited volume. Now, we have always done a 25 microliter reaction, PCR. So we really didn't want to have to increase that to 50. You know, Christine does a 50 microliter because, of course, it's a money savings, you know, by not using so much of your reaction mix and your TAC and so forth. So we wanted to try to keep that volume to at least the 25 microliter reaction. Also at the North Louisiana Crime Lab, we have reduced volume PCR protocols in place, which had been previously validated. So we at least knew that we wanted our input volume to be at least 10 microliters or less with our DNA. So we tried the Lysengo, just like, you know, Christine had suggested. Now, I have to admit, I did not use the Lysengo the same way she did. Because again, we kind of talked and then we kind of started to go different ways. And so what I was doing was cutting my cells into water, you know, however much, at first I was doing 25 microliters of water, PCR grade water, drying off the water, so now theoretically there should just be the cells in there, and then I added like 10 microliters of Lysengo or 20 microliters of Lysengo, depending on if I was going to do a 25 or 50 microliter reaction, putting it in the thermocycler, having it do its thing, and then adding the PCR reagents. Well, I didn't have much success doing it that way. And I sort of abandoned it because we also at the same time were thinking, well, can we just do a preamplification lysis with Pro-K? We sort of do that when we're doing our digest. If we just put Pro-K in there, a dilution of Pro-K, will that, lyse open the cells enough? So we actually decided to try it with a recombinant proteinase K because the thought is that it's a purer form of Pro-K, and maybe since we weren't going to be purifying our sample, that would help.
I just wanted to add something. That if you don't add the DTT to the lyse and go procedure, it will not work at all. And I know you didn't.
Right, and at that point, I wasn't
on that.
Right, I wasn't adding the DTT yet, and that came later. And you can see here in this chart what I did was just quickly plotted doing direct amplification, that 50 microliters of lyse and go procedure, 25 microliter, and this is final volume, and then Pro-K, and then I did start doing Pro-K with DTT. And this was, you can see, this is with 150 sperm. All were 25 microliter final volumes except for the 50 microliter lyse and go procedure that I tried, Profiler Plus, and I had at this point started to do like 30 cycles.
How much Pro-K did you run?
It's a 20 milligram per mil. It's pretty much the similar ratio that you're using when you're doing a regular digest of the Pro-K to the DTT and the volume. So anyway, so as you can see in this chart, the Pro-K and the Pro-K DTT worked the best. What's not reflected here is the Pro-K DTT gave me higher RFUs. So I said, okay, I'm going to kind of go with this and see what happens. So, and you can see we're still using the 377, the workhorse. But I was doing regular and max loads. That's what the two different colors were. Regular load was a 1 microliter PCR product. Max load was 2.5 microliters. So this is kind of what I had come up with. I said, okay, we'll do the preamplification lysis with Pro-K DTT, and that's going to be cut directly into water, drying off the water in a vacufuge, using the recombinant Pro-K with the DTT, lysis time in the incubation, which has stayed pretty much constant while I've been doing this. I do 3 hours at 56 degrees, just like if you had it in your oven or in your heat block for the digest part or lysis part, and then add the PCR reaction components to the tube, and then amplify and analyze. So that's kind of what I had settled on. Of course, we did try to increase our sensitivity because, as you saw, that was 150 sperm. I wanted to see, well, can we increase cycles and get better results? Can we decrease our amplification reaction volume with the reduced volume PCR and get better results? And again, we have in place protocols for 15 microliter final volume of PCR, 10 microliters, and we even have a 5 microliter protocol, but I didn't do that. And then, of course, we wanted to see, how low can we go? So those are different types of sensitivity issues that we wanted to try to address. So this is some of my early stuff. And this is for depiction purposes, 25 microliters, 15 microliters, and 10 microliter final volume PCR. And you can see right away that, yes, the 25 microliter looks good. And all of these 150 sperm gave me full profiles and Profiler Plus. I just put the green here for depiction purposes. But you can see right away that 10 microliter, you're really getting a nice high RFU. One thing I did notice that, and these are all 150 sperm using Profiler Plus, 30 cycles. Pretty good. These all look good. I was getting, anywhere from like 95 to 100% of the alleles called. However, of course, on the end, every once in a while I'd get this just all of a sudden flat line. And I couldn't explain it. Is it because of the way I took the tube off? Did I accidentally touch the liquid and lost my sperm? Did I flick it? Was it static? I don't know. At this point, I still wasn't sure why that was happening. Okay, this is 100 sperm at 30 cycles. So now I started to decrease. How low can I go with this? You can see 25 microliter reaction. I'm starting to lose sensitivity. Had some dropout. And again, with this picture, these are the only two peaks that dropped out of the profile, the whole profile. 10 microliters, I started to get, again, a higher RFU at full profile. So it looked pretty good. You can see there's some stutter there, 13%, 17%. 50 sperm, started to see dropout, but again, I was getting full profiles except for just a couple peaks here and there. So I was excited. I said, okay, this looks like it's working. So up to this point, we could say, yes, we're probably going to be able to eliminate this traditional extraction with the preamplification lysis. And 2, we can get separation of sperm and epithelial DNA with this microscope. So this is a full profile. Profiler plus, 50 sperm, 30 cycles, 10 microliters. 10 microliter final volume. I don't know, this kind of got screwed up, my slides. I was messing around with them a little. And this is 50 epithelial nuclei, 30 cycles and 10 microliters reduced volume. And 25 epithelial nuclei. So yes, we were getting the results too with epicells. So just in summary, I presented at the Promega meeting in 2005, and that's that link on the bottom. I had to actually write a manuscript for that presentation, and if I had known I had to do that, I might not have given the presentation. Because then I had to write this whole thing. I was like, oh, God. So anyway, but that's there, and I do cite references. I do have the reduced volume PCR paper cited, and I also, I kind of took from the way that I started to do this preamplification lysis. It's called Current Protocols in Molecular Biology. It was spring 2005. They actually talk about amplification of single cells with PCR, I think is what there's like a protocol in there. And that's sort of where my jumping off point was with sort of that paper. It kind of gave us some ideas, and that's sort of how we got to where we were. So anyway, we're going to replace the DNA extraction and purification with the novel preamplification lysis technique. We can get physical and complete separation of sperm and epithelial DNA. And at that time, we said, look, the reduced volume, 10 microliter reaction, 30 cycles, with 50 to 150 sperm looked good. Okay, so then, of course, nothing is that easy. And it was literally right before the meeting that I had started to get these troublesome electrophorograms. So I did some troubleshooting. Number 1, of course, is static, and we've talked about that. We had started to see, and even in Louisiana, we can get some really dry winters. It gets pretty dry. And at the time, we had it in this big room, and with air conditioning or anything, even the dry winter, it gets kind of dry in there. And I would notice, these cuttings, we'd cut and they'd be sticking. I'd move to another part of the slide, and here I'd see this piece sticking, or sometimes I even saw the sperm head themselves like popping off, like just popping off even before the cutting was done. And I was like, where are those sperm going? Am I losing sample this way? I'm like, they're so light. Are they just flying off somewhere? Of course, you don't want contamination either. So we did, I talked to Christine again, and she's like, get a humidifier. And, and I'd ask her, well, how much is your, humidity in your room? And anyway, and then another thing is we had a preventative maintenance scheduled, and the engineer came out, and he also said, yes, a humidifier would be helpful. Also, he moved our microscope at that point to a smaller room. So there we could control the humidity. Another thing he said, which I think I kind of talked about in class, the more plastic that's exposed, the better your chance of having that electric charge, happen. So he said, you might want to think about, instead of cutting into 25, can you cut into 50 microliters? And at first I kind of grumbled because I was like, I'm already trying to dry off this liquid. I want this to be a quick procedure, not a longer procedure. And I thought that if I had double the amount of liquid, it would take longer, to dry. So I was hesitant, but I tried it anyway. And sure enough, between having it in the smaller room, being able to keep the humidity at a certain percentage, which, like I said, for us seemed to be about 50 to 65%, worked really well. And then cutting into that larger volume, I mean, really, since then, I have not had the same static issues. It's been much better. So I think those two things really helped a lot. And then I ran up against this electrophoregram conundrum, which is kind of funny, especially me not being a researcher. And, I had molecular biology. I had recombinant DNA, that stuff went out the window a long time ago. I'm like, yeah. So I had these dye-saturated E-grams that I started to get, and so I said to myself, is this contamination? Because that's about the time that Jennifer was doing a lot of her fluorescent research. I said, is there buildup happening on the stage? It's falling into my tube, Because I'd see this, of course, with the 377 data, and it's, fluorescent data. So I'm like, am I having these problems with the fluorescence? Is there some kind of nonspecific binding happening? And so I was talking, I pulled my hair out for like 3 months last summer. Okay, changing this, changing that, what's happening? Why am I getting these crazy electrophoregrams? Well, then I started talking to Jennifer, who was doing her fish stuff, and she's like, well, I'm not getting that, because I showed her, my electropherograms. I'm like, well, what do you mean? Like, she was doing the same procedure I was after she collected her cells. I'm like, well, what do you mean you're not getting this? She's like, look, and she'd show me, her electropherogram. I'm like, what? Well, she was doing Identifiler. I was doing Profiler Plus. I said to myself, that shouldn't matter, So I changed to Profile. I said, well, let me try it with just a different kit, because I thought, well, maybe my kit was going bad. Something weird was happening. Because I'd been using some of these older kits, but they were still not expired, but it was just stuff we didn't necessarily need. So I tried it with Profiler, same thing. I said, okay, something's not right. Well, I did Identifiler. Well, what do you know, it worked. Well, so then I just moved on. I didn't feel very good because I thought, still, I don't know why, but okay, I'm just going to move on. So it forced me to do Identifiler, which is what I wanted to do eventually anyway. So I said, okay, let me just kind of start doing these with Identifiler.
For these peaks.
For which one?
The ones in your electropherogram conundrum picture?
Oh, I took my pictures out. I actually had a picture in there, and some of them were, it depended, like, some of them were really, like, just above noise level, let's say, and then some were kind of higher. It depended on seemed like it depended on if I actually had some true DNA peaks in there. It sort of varied, but they were mostly, like, closer to base level, but a little, sometimes higher, like 100 RFU, 150, 200. It just kind of varied.
Like negative samples and better.
No, because I think I figured out what the problem was. So I'm getting to that.
Well, it looks reminiscent of a problem we had.
Yeah, so maybe we could talk. So anyway, so then I moved on to Identifiler for single amplification, which is one of my goals was to eventually do this anyway. Because if you're working with very few sperm or very few cells, let's do one shot. So that's what I did. So then I went through this process of, optimizing this. And again, because I'm not research by nature, and of course that's why I went into forensic science. It's an applied science. I'm not a researcher. That wasn't my thing. So it was a lot of this, trying to tweak things, not necessarily one at a time. But so one of the things I did was I decided to try to start cutting into TE-4 buffer, and this was because there is, there has been work with reduced volume PCR in our laboratory, and one of the girls that's done a lot with this and the magnesium concentration, she had done a study with TE-4 and Y-filer doing reduced volume, and she found that storage, of course, in TE4 was great for Identifiler, whereas it was the opposite. Storage in water was good for Y-filer reduced volume. We're not really sure why, but so I said, well, maybe I'll just, I just should start cutting to TE4. So I did. I started cutting into TE-4, and then I also even went through the trouble of varying the amount of tris in the TE4, because again, even though this was happening last summer, I started talking to Pat again about this problem with the electropherograms. I'm like, because it started happening with Identifiler eventually. Okay, so again, it reared its ugly head. And so I was talking to Pat, and I said, is it some kind of nonspecific binding? Is there something happening with TAC? I was trying to think, what could be going on? And he said, well, maybe it's something with the pH. Maybe you should try diluting out your tris and see what's, if that's going to do anything. Well, so I did. I, went with the normal amount, and then I put in half the amount, quarter the amount, and a fifth the amount of tris. And what do you know? It was the TE4 and the normal amount looked great. Once I got to having dilute amounts of tris in there, of course the pH changed, and then TAC wasn't able to do its job so well. What do you know? The electropherograms I started to get looked exactly like my electropherogram conundrum. So what was happening was, I think I kept using this old TE4. And I can't tell you how often Pat's like, Kelly, don't use old TE4 buffer. And I'm like, yeah, it's fine, So I kept this old TE4 down there, and I kept cutting into it. And I think at one point, I did change that out, because I was trying to change things one at a time to see what the problem was. But I don't know. I think that's what the problem is. My TE4 would start to get old and break down, and then my pH wasn't so great. And, because we're drying down that TE4. Remember at the beginning, I'm cutting into it. TE4 is getting dried off, so it's concentrating everything. And so if that tris, as tris is breaking down, then the pH is there. It's, EDTA is concentrated. There's a salt, a whole salt issue with the buffer. So I think what was happening is the TAC just wasn't as efficient. Does that make sense? So I think that's what was the problem. So now I remake my TE4 buffer once a week. And I should have listened to Pat. Okay, so of course we did some PCR cycling again. And I tried to look at Christine's paper that she had put out last summer, and she talked about varying the cycle. And we did a lot of playing around, 28 cycles, take it out. Should we add some tack? She was doing the 50 microliter reaction, so she was taking out half, putting it into a new tube, adding some tack. Well, I already had just a 25 microliter volume, so I didn't want to really mess around with that. But we would stop at 20 sometimes, add more, reaction mix. We added more tack. We did these varying little cycling experiments. And then, of course, with low copy number, there's documentation, literature. Some people, like I know the OCME, I'd spoken with Teresa, they're doing 31 cycles. So I said, well, for low copy number, let me try to bump it up one more, because Pat doesn't like us to go up too high if we don't have to, because the higher the cycles, the chances of increased artifacts or high stutter, really looking like a true peak. So we bumped it up to 31, and 31 seemed to work fine. It still does, so I've just bumped up one more cycle. And then, of course, going back to the reduced volume, like I said, we already have a paper out of North Louisiana Crime Lab dealing with reduced volume PCR. And some of the studies have been, of course, because you're drying off that TE4, you want to make sure your magnesium concentration is just right for that PCR, because, of course, I remember from my molecular biology classes, magnesium is imperative to PCR. The TAC, the EDTA in there. So I started varying the magnesium concentration to make sure that, because I was drying off this 50 microliter volume of TE4 and really concentrating the tris and the EDTA, did we have enough magnesium for the TAC to do its job properly? And so I varied, all the way from no magnesium all the way up to, I think it was 2.3 millimole. And sure enough, we found that it's still is in line with our reduced volume work, and that you need about half a millimole to 1.5 millimole extra magnesium added to make sure that your reaction is optimized. Yes, that's when you're drying down that TE4 in the vacufuge. So I'm sorry about my slides, I got a little screwed up. But so this was before I finished optimization. This was 50 sperm with identifier at 30 cycles. You can see I'm seeing a little dropout, but still it's not too bad. I was getting 24 of 29 peaks called. This is after I optimized the magnesium. The TE4 was good, 31 cycles, and you can see I got 29 out of 29 peaks. That made me so happy. You can see a little bit of stutter here. I'm trying to think. I can't really see what it says. Not bad. So I was very happy with that. So the next question becomes, well, how many sperm do you need? How low do we need to go here, or how low can we go? So again, here's this 50 sperm with identifier. Okay, this is 25 sperm. This was actually before I had finished my magnesium optimization. 25 sperm, I was getting 23 out of 29 peaks called. But the only thing, of course, about this was with the 25 sperm, it wasn't always reproducible like this. Sometimes I would get this nice profile. Other times I just get sort of a partial profile. After I optimized, I just ran some more just last week. This is 25 sperm, and I was able to get 27 of 29 peaks. But again, our target seems to be 50 with this, 50 sperm. The 25 isn't as consistent with getting these nice full profiles. I mean, we still get peaks. It's not always so, full or complete. That could be just a collection issue. That could be, is the sperm I collect, am I not getting them all? Am I getting sperm that are already starting to degrade? I'm not sure what's happening, why it doesn't happen every time. But 50 seems to work well for us every time. Here's a 15 sperm identifier, and again, before I had optimized, here's 15 after I optimized. So even with optimization, you can see a change. But then, of course, the issue becomes, at what point with a slide, if you don't have enough, if you don't have 50, at what point do you then collect the 25 or 15 that are there? Or do you always want to be able to amp 2 tubes with identifier? How many people do identifier in this room? Do you do it all the time with low copy number?
We do 3.
3 amps. Okay, what do you do it on?
We don't do low copy number.
Okay, how do you use your identifier? Like Casework, or do you do it with references?
Yeah, both.
Do you always duplicate your identifier?
Not
Just one amp? Is that what you?
We duplicate like it was same rules as when you were on the OCV. If it's probative or a mixture, we dupe it.
Yeah, okay. See, it's been a while since I'm like, after doing it a certain way for so long and then you go someplace else.
Even if it matches the victim, if it's a probative sample, we'll duplicate it.
Right.
And if it's a mixture, we duplicate
So in doing a single amplification, how do you confirm, or you know what I mean? Like, how do you confirm that that's your result and not a mistake or something? Because that's always my thing with identifier. With one amp, I say, well, gosh, how can we show that this is in fact reliable in it? You know.
Just be careful.
Yeah.
Controls and the whole setup. You know. The
If you
negative amp,
have,
positive amp.
let's say if you have an exclusion, like let's say it doesn't match your victim, it doesn't match your I mean, is it always matching somebody? And if it doesn't, do you do something, or
No.
do you look at tables when you've got all your PCR results from a set of samples, or?
Well, as an analyst, if you're batching your cases and the evidence, let's say, excludes the suspect in the case, you'll look at other samples in the run just in case.
To make sure, yeah. Because that's always something I'm thinking about. I'm like, okay, how will we do this? But, right, so that's something that is one of those case for considerations that now have to be worked out. Do you cut 2 tubes always, 2 tubes of 50, 2 tubes of 25, or 3 tubes of 25,
Interestingly, though, that's only we're a Promega lab in PowerPlex 16. We've always done single amps.
Yeah.
And part of that
It's
thought
the
process
thinking.
came from the concordant loci, I think,
Mhm.
where you guys wanted to be
Yeah.
able to check that
Yeah.
you had the right, there was no tube switch.
Right. Yeah, it's hard to change your thinking to say, well, do we really need to do that? Is it fine just to do one and just be happy with the results? Yeah, no, I can completely see what you're saying. It's hard to.
Isn't it just also, I mean, I think a duplication are not just, it's one more time. Like, what if there was a switch in the first amp or something? It's just checking. You're saying this profile matches your suspect. It's just
Right.
checking that you didn't make a mistake. And I think once you delve into the smaller numbers of sperm, you have to do replicates.
Oh, I would agree with low carbon
So-
reading to do replicates, but for just normal casework samples,
Yeah.
for stain level DNA, I think that's what your controls are for, your reagent controls, your amp controls. That's what those tell you.
Yeah.
How do you know there was
You're not
no
switching.
mix-up anywhere else?
Sometimes you only have enough to do one amp.
Yeah. What if you mix it? You can mix it up twice. How many replicates do you do?
Difference of opinion. I mean,
Yeah.
I
think the
feel
thought-
comfortable too.
I find that interesting, the thought process, because you had with the two different, the profiler plus and the co-filer, you have that built-in,
Overlapping.
and then it's been a nice way to compare, but then and you've gotten used to that. But where along the whole process, maybe in the early process, you can switch samples to there, but is there any double check there?
No, that's what
Are
I
you
mean.
making 2 cuttings? Are you making 2 swipes? I'm just kind of
But I think
devil's
we
advocate.
try to do the best you can. I mean,
Yeah.
another way to look at it is to do it once, and that if it hits, duplicate it. Only duplicate ones that are, that you're putting someone in quote unquote in jail for or something. I mean,
Yeah, see, the OCME too is a little unique. I mean, I know now like New York State, there are some large labs now, but some of that is too because so many people are handling those samples. I know they want to be sure. Look, we want to confirm this and make sure there wasn't a mix-up. Because so many people are touching, a given case. That's
so
fine.
not just
I know
one
that's part of
analyst.
it.
Correct. It's several people
Yeah.
who are involved.
Because I remember when I was there, they actually would go through the process of, duplicating a reference, and a separate person would go cut that card. Is that what they still do?
Yeah.
Right. Because they had someone who would go back to cut, and it was the same person. They were pulling like a 2005 bloodstain card instead of the 2006 or something, and then so that was actually it was a longer time ago, but that was why they went to that. So
Are all your samples profiles searched against the staff database?
what do you mean like
Any-
a local
any
database?
Generated profile, you
Yes,
search against your staff
yes.
database? That would be that's how we make sure
Right.
that there's no
But you can
contamination.
have sample to
To
sample.
switch
mix-up.
cases
Sure, but you can
if
have
you're
that.
batching.
You can have that at the envelope stage or the crime scene stage. There's
But not if you only open up one case at a time on
yeah.
the bench and only have one thing open at a time. So it's just a difference of approach. And I think in New York we're very conservative. Maybe the atmosphere where we are, the state where we are.
I think
Well,
it's
and it's-
really primarily how you guys are handling casework,
Yeah.
because every laboratory has different
view.
staffing, different procedures, different methodology. And the good thing is that there are any number of ways that generate perfectly reliable results. And as long as it works for your lab, it's good for me.
Right. I think again it's sort of a batching issue because like at our lab, I was telling some of the people at lunch, we're almost a novelty now. We start from beginning, work the case all the way through end, one person, so of course issues are different there, whereas if you're batching, you have lots of different people touching things. So I think that's part of it too, like a difference. But anyway, I was just curious. We could talk about it more at roundtable if you guys want. So anyway, so we've sort of settled, like I said, on 50 sperm. I'm still going to probably do a little more of the sensitivity of how many sperm, since I have this optimized finally. But the 50 sperm out of the last, since I've optimized it, I'm getting a profile every time. I haven't had a dropout again. I haven't had that flatline happen. I'm definitely going to be doing more. And I've gotten I think it's around 98%, 99% allele call with the 50. So it's really working. I'm excited. So what happens if we have no sperm observed? Now, of course, my thinking, Pat, with research and validation, he just wants to keep going on tangents and he likes research. He keeps thinking of things, and I said to him right away when I started this, I said, Pat, I just want to at least get this online for basic separation. Let's not worry about if there's no sperm, or let's not worry about fluorescence just yet. I want to get this basic procedure online. But still, if you're spotting a large amount of your pellet or most of your pellet onto the slide, well, what if there isn't any sperm, but you have PSA positive? Well, what do? If we're not doing fluorescence yet, what are we going to do? We have to do something. So we said, well, no sperm observed. Well, gosh, the great thing about these pen slides is they're no longer it's not your sample inert on there. We can take a disposable scalpel and cut out that whole spot. That's what I like about it. That's why I don't want to get rid of them. Cut out that whole spot, and we can just extract it. Because at least maybe we can get some useful information from maybe YSTRs, So that's what we did. We did a side-by-side study where we pelleted our material, took half of it, spotted it on the slide, and left the other half in the tube, and then did a side-by-side, comparison of cutting the spot off, extracting it with organic, or just doing a regular organic of the spot. And there was no difference in the yield, no difference in the STR profile. So at least for now, I think before we even get fluorescence on or see if the fluorescence is going to work, to me, if we have very, very low number of sperm that we see on a slide or no sperm, at least we can cut this spot out, put it in a tube, and extract it.
So when you say there's no difference in yield, then are you saying that you had 0% loss from the
well, there was no significant difference, like with the t-test, as far as like our peak heights. There was no significant difference in the results of the two compared, side by side. Quantiblot, like not Quantiblot, but real time.
So you lost nothing in the laser microdissection then?
No, this is cutting out the spot with the scalpel.
Oh.
Yeah, we actually physically took a disposable scalpel and just cut the spot up there and stuck it in a tube.
This was just a test to make sure that if you do put it on the slide, you haven't lost anything. You could have left the pellet in a tube or put it on the slide, and in that instance, you haven't lost anything.
Okay.
Well, and kind of like what Andy was saying, he said, this foil is biologically inert. I just wanted to make sure that the foil actually didn't cause any problems, So this is an example. The girl that helped me with this, she made varying ratios of male and female epithelial cells. And of course, here you see we have a 1:20 ratio of male and female epithelia. You don't, with regular SGM plus panel, you don't see a Y. It's all female. But sure enough, even with the foil cut out, we're still getting a full Y profile. So that's what's nice. At least if at this stage in the game, if we decide to put this on for a regular, just separation of sperm and epithelial, we've made the spot and we don't see any sperm, or we just maybe see a couple and we say, well, we can't cut these out and do anything with them. Let's try, regular extraction and typing. At least we can get some kind of information, hopefully, from that spot. So some other advantages of LMD, which we've sort of talked about, but not really. Number one, if we're able to get the absolute separation of sperm and epithelial DNA, and we're cutting, remember, we're just cutting very specific cells out, does that mean the effects of traditional PCR challenges are going to be minimized? And then lastly, of course, if we're able to eliminate the traditional extraction, if we eliminate the quantification step, look, we're hopefully going to decrease analysis time for a sexual assault sample. And we're kind of envisioning this novel process that we're going to use, and Pat will go into that more. So what I meant when I said, are we going to minimize the effects of traditional PCR challenges? Well, number one, it seems very clear, but if you're simplifying your mixtures, the great thing about this is you hopefully will then simplify your interpretation. For your final report, hopefully there won't be this whole, got to deduce major, minor, and I have, you know, the vaginal wash or vaginal swab, you know, oral swab, you know, gave me mixtures, and you got to do the statistical analysis, and you know, whether it be likelihood ratio or you know whatever. So hopefully you're going to simplify your interpretation, thereby simplify your statistical interpretation, which will be great, of course, for your report, great for court. So these are some of the advantages too. Of course, we've hit on this a little bit. Because you're not collecting that whole sample, are you then minimizing your chance for contaminants in the sample itself? Minimizing your chance for collecting inhibitors. So those are things that are kind of nice. And then, of course, we've already gone through this, increase your PCR cycles to enhance low copy number sperm. Because you're not capturing those contaminants, hopefully, and you're not capturing those inhibitors, then maybe that's why we're able to increase the cycles without much difficulty. So, of course, we envision a new way of processing and storing our sexual assault samples. We're going to be storing them on our slides instead of in tubes, and hopefully this will be a faster process. And like I said, Pat will go into more detail. I just did want to show you this slide just to give you an idea, at least of this process that I'm doing right now for the validation. Of course, you're going to do still your presumptive testing, and depending on the place, you know, it might be a little different. But for us, we're doing AP testing. We do the ABA card. So I estimated about 2 hours. You know, spot your slide, do your staining, and this includes, of course, we do a 1 hour, you know, about a 1 hour shake of your swab or your cutting. Of course, you're going to examine for sperm and cut at the same time. So as soon as you find you have sperm, you're going to assess that slide. You're going to take your cuttings. And as you've seen, yeah.
Just PBS, you said?
Yeah, just a 1x PBS, you know, to make your pellet. So shaking. So examine for sperm, and with our old microscope, I estimated on a, you know, and these aren't heavily populated sperm slides, just a good population. It takes me about 15 minutes to cut, and our microscope is much slower. And then I went up to an hour, and that would be if you had a difficult slide, you know, if you're collecting a lot of epithelial cells, let's say. So I think the estimate of an hour is actually a pretty, you know, conservative estimate. It's probably going to be faster. But even then, an hour to cut your material, I mean, that's great compared to a differential, organic differential, no less. So drying of the TE4 actually doesn't take me that long. It's only about 45 minutes. And I think it's just because there's not a whole lot of nuclear material in there. So it actually dries off pretty quickly. And that's drying in a vacufuge at about 30 degrees Celsius. I do my preamplification lysis in the thermocycler, and I usually do that overnight. It's about 3.5 hours. Like I said, I do 3-hour incubation at 56 degrees, ramp it up to 94 to help denature any proteins, the pro-K. And then hopefully the next day you could do your PCR and your gel. 31 cycles takes about 3.5 hours. And then, of course, if you have CE, it's even faster. The great thing I wanted to point out about this, and I've been doing this for a long time, this process, not only does it improve and streamline the analysis of sexual assault evidence, but it frees up analyst time. This is what I loved about this, because a lot of this stuff that I'm doing is just a quick addition of reagents, and then it goes in the thermal cycler for 3 hours. And then I take it out, I add additional reagents, it goes in the thermal cycler again for 3.5 hours. The drying is 45 minutes. So really on the front end is your most labor. You know, your screening and your cutting. But after that, it's nice. Like I can throw stuff in, go do something else. You know, go do my paperwork, my casework, or whatever job I'm trying to do. So that's what's nice. That's what I like about this procedure. I feel like it frees up a lot of my time. So what are some of the considerations I still have to do? Well, I'd like to see if I can shorten the lysis time from 3.5 hours. Again, I kind of went from that current Protocols in Molecular Biology article, and I think that's where I came up with this lysis time. So I want to see if maybe I could even shorten it to an hour. So that's something I still want to play around with. I really want to do a full-blown mixture study, kind of like what Christine talked about, and this is mainly because of that novel validation. I've kind of been looking through the validation. You know, I try to keep revisiting the validation guidelines to make sure I'm not going to forget doing something, and so I want to do a little more, a better mixture study. The epithelial nuclei, same as Christine, I don't have quite the same success with the epithelial nuclei. I get more partial profiles. You know, even with more, like if I double, let's say 100, or no, 50 epithelial nuclei should be about the same as 100 sperm. For some reason, I just don't get the same, and I don't know if it's because it's not so pure, you have a lot of nucleases floating around. I mean, I'm not sure what's happening. I don't know if there just needs to be a purification to happen if we're going to do the epithelial nuclei. Of course, one of the things we thought of, we said, well, do we need to do epithelial nuclei? Well, that's kind of one of those things. I think for an intimate sample, I think the only instance I could see it being important is just to confirm that that swab actually came from that victim. Because we know that can happen sometimes at the hospital. I know that happened before at the OCME where the swab was not matching the victim type, the epithelial fraction. What do you know? It done again. Well, sure enough, what happened? There was a switch at the hospital. Wrong swab, wrong box. So, I don't know if, you just need to do at least, one panel. Also, if you had just regular evidence, like a sheet or something, yes, you'd probably want to do your epithelial cells. So that's still something I have to work on. Obviously, do some non-probative samples. And then lastly, which might take a while even, is just the considerations for casework. For example, how are we going to assess the slide? At what point do we say, okay, we have enough sperm or we have enough epithelial cells? Do you count every field or some fields? Do we have to do the quantification? How are we going to process our blanks? How are we going to process the positive control? Are we going to batch cases? Can we batch cases? Do we have to clean the slide holder? Do we have, there's all these kind of issues, of course, that have to be considered before you can actually implement.
There's still more there. Do you keep going or do you stop at 50?
Well, and that's another casework consideration. That's something we've talked about. I mean, if we have more, should we do more? But, I actually found with this 50 sperm, I'm getting such high peak rates. I'm averaging, let's say, 500 to 1,000 RPU. Yeah, I think it's going to overblow is what's going to happen. So, yeah, at least you want to make sure you have enough on that slide that if you had to go back, or something, cut 2 or 3 tubes, or, there's got to be something. Some of these considerations, of course, have to be hammered out. So I can see even that part I can see taking a little while.
Is it more stable to leave it on the slide or to cut the second tube if you're storing for later duplication?
Yeah, that's a good point. You're talking about if you were to go back later.
Right. That's like you decide not to duplicate at that time, and you're going to have a policy out just to dupe later. Is it better to leave it on the slide and dupe if necessary, or to cut it and save it in the tube?
And that's a good point. Or at least cut it 2 tubes and just do them. Or cut it, like sometimes what I would do is I would cut them, dry off the TE, and stick them in the freezer that way. Is that stable because it's dry? But that's another thing. Stability is one of the novel validation things, and I think I'm going to have to do a study of the slides themselves, 6 months out, maybe a year out. At our lab, we have a policy of after 180 days, and we put this in our report, after 180 days, we throw out pre-amplification work product. So they have 6 months to be able to come, take that if they want, have someone else test it or whatever. So of course this slide would be pre-amplification work product. Do we throw it away? Are we going to just do stability study based on that 6-month window and then not worry about it after that? Yeah, I mean, it's like a Pandora's box. So.
Kelly, I just wanted to add that by comparing my study and your study, just you said it, that we both had trouble with the profiles for the epithelial cells, and we both used non-purification extraction methods. And then when I used like a QIAGEN cleanup method, I had much improved results. So it's something you have to consider, that when you're dealing with epithelial cells, you might have to have a different extraction method.
Yeah, and I think I may try the QIAGEN method and just maybe we'll have to do something like that with epithelial cells. So.
I'm curious, did you ever take 50 cells and use your extraction protocol and take it into QIAGEN? Maybe I missed this. If I did, I'm sorry.
Oh, that's
and
okay.
Did QIAGEN extract on those 50 and just compare the results?
No. You're saying try the QIAGEN procedure versus just cells. My procedure?
Yeah.
On 50
I was just
sperm?
curious if you ever tried
No, I
that.
have not done that yet.
Yeah.
And I may try that just to see, especially if I'm going to do it on epithelial cells, I'll probably do it with the sperm too. But, because I kind of got off on this track and I just kind of wanted to keep going with it. Because, if you try this and this, it's going to take you forever. Yeah.
And what DTT concentration are you using?
I think ours is a 40 millimolar. I think she said hers is 30 millimolar. Ours is 40 millimolar. Okay, well, before the when Christine, asked me about doing the class, she said, have you done any casework examples yet? And I was like, no, I've been trying to optimize this identifier. I said, I really want to try some, but I just haven't had time. And she said, well, do you think you'd be able to? I said, well, you know what? I'll try to do something before the class. Well, it ended up I was doing the casework examples last week. So all I did was I grabbed 4 casework swabs. 3 of them were still sort of ideal type of samples. 3 of them were old proficiency kit swabs that I took. So of course we know these a lot of times don't have a lot of epithelial cells. But I just wanted to say that up front that these are sort of, they're casework examples. They're a little more ideal, but I just wanted to kind of show you what I got. The 4th one was a true sexual assault kit, like Pat was talking about, a kit that was taken from somebody, and then charges were not, pressed against the suspect or whatever. So they said you can do whatever you want with the kit. So the 4th one, actually, it's case number 1, and I'm not starting with that one. It was a true type of sample. So, and this, I didn't go into as much depth as Christine, because again, I was kind of doing this last minute. But so here's the first one. This is case number 2. This is 50 sperm with identifier, doing the procedure I talked about, 31 cycles, with the improved magnesium concentration and the nice T4. And this is what I got. Okay? Now, in looking at this profile, at first I said, what's going on here? Does it look like a mixture? Well, not really. Of course, you do see here, this peak here, this peak here. Actually, maybe I'll use this pen. I've labeled those as stutter. And that I just did, again, blind. I didn't know what the results were. I didn't have my old proficiency test results in front of me. I pulled up these electropherograms and looked at them. And I did edit stutter. Now, these are the only 3 loci, of course, that look like there's 3 peaks. And these were all in stutter position. So I felt pretty comfortable. Clearly you see here. Let's see. Oh, crap. This loci, this locus is pretty strongly imbalanced. And the peak, of course, the stutter peak isn't much lower than the true peak on the right. But just by having done a lot of cases interpretation, I said, well, this is more likely on the left to be a stutter peak than the one on the right. So I just said this to me is probably a stutter peak. Fairly high. Again, when you're working with lower copy number, this is about 150 picograms, you're going to see this imbalance from time to time. But anyway, that's what that is. But so anyway, then I went to the case. I pulled out my old information. This is how we make our tables or charts, as they call them in the South. We call them tables at the OCME. On the left is my DNA profile from the male from my proficiency test the first time. And then I cut 2 tubes because I had there were plenty of sperm. So I said, let me just do 2. I did 2. Any little editing I did, like I showed you, was blind, and these were the results I got. Full profiles. They matched my old profile, so I was very happy. At least this is working. I mean, yes, these are a little more ideal world samples, but at least this is working. This is working on just a sample I pulled out. Case number 3, same thing. This is there's a stutter peak right there, but otherwise this looked great. And again, I cut 2 tubes of 50 and made the comparison, and again, I got full profiles. They were the types I expected, and it seemed to be working. And not to beat this overhead, but here's case number 4, same thing. Looks great. I mean, full profile. I was so excited about this. And again, you're seeing some imbalance, but again, not to be expected, with low copy number. And reduced volume PCR. Okay, so case number 4, again, cut 2 tubes. They gave me results. They matched my old results. So I was really, really happy about this. So this looks good. Okay, so case number 1, of course, was the true case. And what do you know? This is what I get.
How many cells did you cut on the proficiency cases?
50 sperm. Mm-hmm. Just as I've said, is about our target. We think this is going to be our target amount is 50. Okay, so we're seeing a mixture at least, three loci here. And it didn't surprise me because of the slide, but I'll show you that in a minute. But it still looks good. I mean, I'm still getting good peaks. If anything, I was having a problem a little bit with my migration through the gel. The peaks at the end are looking a little wide. They looked kind of crummy, but I didn't have time to rerun. So anyway, so case number 1, I pulled out the old results. And this, again, this was actually a girl that was training, had done this previously, and that's how I knew this kit was positive for sperm. She had trained, and this was one of the kits she used, and she only did the SGM Plus panel. So I had done identifiers, so I don't have the profiler results. But anyway, I did pull out the epithelial fraction and the sperm fraction types, and at least, even though I was getting a mixture, I was happy that I wasn't getting any foreign peaks, like any peaks I couldn't explain. The peaks were all either from the male or the female, they could be accounted for. So again, I still was getting a fairly good profile, but I had a mixture here. Well, let me show you the slide. This is what it looked like. This is what at least when I put my pellet on there, this is what it looked like. Well, you can see, I mean, there's a lot of what I call, cellular debris, a lot of this stuff. I mean, it was actually a little hard to, pick out the sperm because there was so much stuff on here. So a case like this is another one of those casework considerations. Are we going to have to, just before any case is spotted on a slide, should we do some type of, like what Christine was saying, a mini lysis or something to help get rid of any extra female or any DNA that's just kind of floating around there? Because probably what's happened is, I'm guessing, cells that are degrading or whatever, and they're having a affinity for the sperm or whatever. So I think that was why I was having a hard time separating, because both my tubes looked like that. If you saw, I had both gave me pretty consistent mixtures. So anyway, so that's sort of my little casework thing.
This sample had a lot of sperm.
This?
Yeah, this.
Yeah, it did.
So the regular diff worked great. And
yeah. There were plenty of sperm. It was great. But obviously, kind of like Christine, she had one case where it didn't work as well. Same kind of thing. I obviously had a situation here where there was a lot of just, cellular debris is what I call it, just floating around on that slide. So I could not seem to get those sperm free of the DNA, extra DNA that was there. So I don't know if there's going to have to be a dilution of the pellet, if you're going to spot a small spot and look at it to kind of get a feel for what your sample looks like, So, and then I'm sorry?
Are you staining those cells? I
These?
may have missed that at the beginning too of your protocol.
Oh, I'm
Are
sorry,
you
yes.
using
I
the
took the same, I took off from Christine where she had done the staining studies. I didn't do any staining studies again, because I just didn't want to reinvent the wheel. I wanted to, working in a crime lab, I said I just want to get.
Just for the initial assessment though, like when you were selecting your sperm, did you stain your slides?
Yeah.
Was it Christmas tree
You
stain? or
mean
was it
the old
H&E?
slides?
No, just for these.
Yeah,
like,
initially I
just all your
did
studies
Christmas
and that.
tree. Yeah, initially I did Christmas tree. And then at one point I talked to Christine and she was like, yeah, with the Christmas tree staining, I've noticed that green may, slightly inhibit because the peak heights were lower. And she told me about her short nuclear fast red staining. I said, oh, I might as well just do that, And I may have even seen her talk, because I've seen her talk a couple times too, so. Yeah. And then the rest of the slides on here, this is just stuff I kind of stuck on there because you guys are more managers, technical leader types. I thought we could have a discussion about validation if we wanted or whatever, but we can leave that for roundtable. But I did stick in here, if you wanted to get into validation, I pulled up Promega's Profiles in DNA article from September 2006. I don't know if any of you read that, with John Butler. He was a member of the SWGDAM validation subcommittee who helped revise the validation guidelines. And I thought he made some really good points in there about, obviously, and he's talking more about an internal validation, but if you were to take some of our ideas or whatever, go back to your lab and say, okay, we want to do a validation or whatever, I think that's a really good article just to kind of help keep you focused and not doing too many things.
When you're on the cusp of LCM, I think 5 is a great number for high copy, but not for low copy.
Yeah,
It's
yeah.
too low.
And you're right. And Pat's the same way. He's like, sometimes it's just not enough. You got to do more. And I sort of sit in between where I say, you know what? I think you could you can validate a procedure and at least get that procedure online, and then if you want to do something like fluorescence or whatever, kind of then you can continue to do that type of validation and then put that online or whatever. I mean, I don't know. But again, I haven't done a whole lot of validations myself. So, but just because we're in crime labs and we don't have any time, it just, if there was a way to do these things a little quicker. But, and then I threw his suggestions in there just in a summary. But again, I have another link down there, which you can't see. Well, I guess you can see it okay. But anyway, my acknowledgements, of course, are, Pat, my boss at the North Louisiana Crime Lab, and of course the lab itself for even buying the instrument. A lot of the people in the DNA section helped me in some small way or another, whether it be cutting cells for me or, helping me with the magnesium concentrations. And then, of course, Christine guided me a lot at the beginning, and Andy has given us some free slides to do work on. So that's it. Does anyone have any questions? Curious about anything I didn't go into? I mean, this is very, brushed over of everything I did. Yes, I'm hoping to write a paper, right, if I ever find the time. But anyway, yes. So, no questions? And if you guys want, we can talk about some of this other stuff. I wrote a whole list of things we could talk about as far as casework considerations. I mean, so.
Kelly.
Uh-huh.
At this point, you're not looking at fluorescence. They may not understand, but you were talking about negative slides up there once. But right now, you're only looking at stained material. You're not looking at any fluorescent material or anything like that.
This is all the stuff I did was with this nuclear, for the most part, just the nuclear fast red staining. Some of the fluorescence that they did last summer was, like I said, with the procedure I had started to do, but again, it wasn't optimized. But yeah, I haven't done any fluorescent staining of sperm or tested those. Because again, I was trying to stay as focused as possible. So.
About with your troubleshooting on the TE4 and talk about nuclear fast red, it reminded me something that we noticed in the last class. When the last class prepared the nuclear fast red slides and we started looking at them, we said, wow, they look really good compared to what we have.
Yeah.
It's really nice and intense. And it's kind of one of those things, again, it's like using that same old bottle.
Right.
I think, after a while, you don't realize slowly you're losing that intensity staining.
Yeah,
so something to consider when you're using now. Before maybe, you have your Christmas tree stain, you're doing thing, your bottle is a year old, and you don't think about it because you just want to quickly look at sperm. Something to consider, especially since, microscopy is such a crucial role in this analysis.
Yeah. I'm actually going to see about getting some of that Visus nuclear fast red. We have Fisher. And still, even with the new bottle, I just feel like it's not stained very intensely. Of course, it is a shortened time, but the Visus stain in there under,
Vector?
Or is it Vector? Sorry, Vector.
Yeah.
It's just 5 minutes stain. It looked great. So I was like, oh, we're going to get some of that. So, but that's all I have.